Have you ever wondered why human populations around the world exhibit such fascinating differences in their genetic makeup, even while sharing a common ancestry? It is a truly remarkable aspect of our species. The variations we observe in human genetic patterns, known as population structure, are primarily created and maintained by four fundamental evolutionary forces: gene flow, genetic drift, natural selection, and mutation. These factors constantly interact, shaping the genetic landscape of human groups across generations. Gene flow, essentially the movement of genes between populations, happens when individuals migrate and interbreed with new groups. Imagine people moving from one village to another and having children. Their genes mix. This process tends to make populations more similar genetically over time by sharing genetic variations, reducing the differences that might otherwise accumulate between them. So, high gene flow means more genetic blending. Genetic drift, on the other hand, is about random chance. It is particularly powerful in smaller populations where random events can cause certain genes to become more or less common purely by accident, not because they offer any survival advantage. Think of a small group of people leaving a larger population to start a new settlement. They carry only a fraction of the original gene pool. This founder effect can lead to unique genetic signatures. Another example is a bottleneck effect, where a population dramatically shrinks due to a catastrophe, and the surviving individuals carry only a limited subset of the original genetic diversity. Genetic drift, unlike gene flow, tends to increase genetic differences between populations. Then we have natural selection, which is a powerful mechanism where individuals better adapted to their environment are more likely to survive and reproduce, passing on their advantageous traits. For instance, populations living in areas with high levels of malaria have often developed genetic resistance to the disease. This is not random. It is a direct response to environmental pressures, leading to adaptations that become characteristic of a particular group over many generations. Natural selection fine-tunes populations to their specific surroundings. Lastly, mutation is the ultimate source of all new genetic variation. These are random changes in our deoxyribonucleic acid, or DNA, that can introduce entirely new traits into a population. While most mutations are neutral or even harmful, some can be beneficial and, if selected for, contribute to the diversity and adaptability of a population over evolutionary time. Without mutation, evolution itself could not occur. Understanding these forces allows researchers to reconstruct human migratory paths, identify ancestral connections between groups, and even pinpoint genetic predispositions to certain diseases that might be more prevalent in specific populations. Scientists analyze genetic markers, like single nucleotide polymorphisms, or SNPs, to measure the extent of these factors within and between human groups, building detailed pictures of our shared and unique genetic histories. A crucial concept in this field is the Hardy-Weinberg principle, which describes a theoretical population that is not evolving, meaning these four factors are absent. Any deviation from this equilibrium signals that evolutionary forces are at play, providing a baseline to quantify the impact of gene flow, drift, selection, and mutation. This principle helps researchers dissect the specific mechanisms driving observed population structures. It is also how companies offering ancestry services can trace your family's geographic origins with remarkable precision, by comparing your genetic markers to vast databases of populations shaped by these very factors. Ultimately, recognizing how gene flow, genetic drift, natural selection, and mutation interact provides us with a profound framework for appreciating the intricate tapestry of human genetic diversity. It illuminates the journeys our ancestors took, the challenges they faced, and the deep interconnectedness of all human life, revealing the dynamic story of our species imprinted in our genes.